We have Dr. Judson and Dr. Hunt they're going to be talking about UBE3A overexpression mice and a collaborative project they've been doing in their institutions. And like I said, it's very, very late for one of the mats, so we'll get going.
Okay. Can everyone see that okay?
We can.
Okay. All right. I'm going to get started then. And I just want to start by saying it's absolutely an honor to be speaking to this audience about our recent efforts to develop mouse models of UBE3A overexpression. And just want to emphasize that these models are designed to lend insights into the pathophysiology of Dup15q syndrome. So think of them as overexpression models for Dup15q syndrome and not mouse models of the disorder, which would involve a lot more genes and be much more complex. So before I get started, I'd just like to thank the organizers for the opportunity to speak and also to acknowledge key colleagues and collaborators, notably former and current Philpot lab members, including Brittany Williams, Mike Sidoroff, and Courtney Davis. And of course, Matthias from Ube Garisma's laboratory in the Netherlands, with whom we shared the model and whose excellent work you'll hear about shortly if he doesn't fall asleep first. So he's been quite the trooper this evening. So finally, I'd be remiss not to point out very generous funding from the Dup15q Alliance, without which this project wouldn't have gotten off the ground. So without further ado, I'd like to jump into this next slide. And as you just heard, Dup15q syndrome results from duplications of a critical region of chromosome 15. And what's immediately obvious from this simplified schematic is that this region encompasses a lot of genes. And this is daunting when you're trying to understand how overexpression of any one of these might alone or in combination contribute to disrupted neurodevelopment. But we know that due to the significance of the maternal inheritance of these duplications that we just heard of, there's a spotlight on one gene in particular, and that is UBE3A. And it's the only gene in the region that is expressed exclusively from the maternally inherited allele in mature neurons anyway. And it's the one whose overexpression we chose to focus on in our mouse model. So one gene to shine a spotlight on and to start with to gain some traction into the pathophysiological origins of the disorder. So from the outset, we wanted to maximize the utility of our UBE3A overexpression models. First, we wanted to ensure that the overexpressed protein would be functional, as it presumably is in the bodies of individuals with Dup15q syndrome. And this means not attaching anything extra to it that might affect where the UBE3A protein travels within a neuron and how it might work whenever it gets there. And second, we wanted to recapitulate disease-appropriate overexpression. And this means making a mouse model with exactly 1 or 2 extra copies of UBE3A, akin to what's most commonly observed in interstitial Dup15q or IDIC15 cases, respectively. It also means ensuring that the mRNA and protein expressed from these extra copies goes to the right places in the brain at the right time. So we made this all happen by basically hijacking bacteria to grow a large piece of mouse DNA that encompasses the entire UBE3A coding region, which is schematized here, and importantly, these very large upstream and downstream sequences that presumably house all the necessary instructions for properly expressing UBE3A protein. So after a few subtle tweaks that could help us out in future experiments, but that I won't belabor at this time, we harvested this DNA from the bacteria, we purified it, and then we set out to make a mouse. So how did that work? Well, we accomplished this by injecting hundreds upon hundreds of fertilized mouse embryos with this purified mouse DNA that I just told you about, with the hope that some of them, in some of the mice, an intact piece would land safely somewhere in the mouse's genome. And once those embryos grew up into mice, we could screen them to see if they indeed had received the extra UBE3A copy that we delivered, and if so, how many of them they received. So importantly, through a little genetic trickery that, again, I won't have the time to go into detail about here, we were able to take UBE3A overexpressor mice, founders that we determined to have multiple extra UBE3A copies, most often 2 extra copies, and create from them sublines of mice with just a single extra copy. And this kind of subline variegation, as I like to call it, was critical for some of the behavioral experiments that Matthias and I will tell you about in just a bit. So what you're looking at in this next slide is basically an example of genomic validation of our principal UBE3A overexpressor mouse model, which we've been working with in earnest. And it happens to carry 2 extra copies of the mouse UBE3A gene. So specifically, this plot shows signal for UBE3A specific sequences across the entire mouse genome, which is arranged, if you look at the y-axis here, by chromosome. So X here at the top, all the way down in descending order to chromosome 1 at the bottom. And if you look out along the x-axis, you can see peaks of UBE3A signal. And of course, there's one on chromosome 7 where the endogenous mouse UBE3A gene resides. But there's also conspicuously 2 very strong peaks located adjacent to one another on chromosome 3. And this signals the location of 2 extra transgenic copies of UBE3A that we introduced through our transgenesis experiments that I just described. So we confirmed this using a complementary qPCR approach shown over here, which clearly showed a doubling, confirmed a doubling of the total UBE3A copies in this line. So to determine if any of these extra UBE3A genes actually increased UBE3A expression, we wanted to probe for UBE3A mRNA levels in the brain. And as you can see on the x-axis, mice of various genotypes are arranged according to the number of UBE3A gene copies that they express in neurons. So kind of the active copies when you consider the paternal imprinting that's going on. So for example, due to silencing of the paternal wild type allele, wild type mice are expected to have 1 functional neuronal copy of UBE3A, shown here. Whereas UBE3A+1 mice with an extra copy, or UBE3A+2 with 2 extra copies, or UBE3A+4 with 4 extra copies, have in their neurons a total of 2, 3, or 5 extra copies, or 5 total functional copies, as the case would be. So, and if you look at the y-axis, you can start to look at how the mRNA expression is plotted. And of course, you're seeing clearly mRNA levels increasing as a function of UBE3A gene copy number. But what's notable is that this rate of increase is somewhat less than we would expect if it was a purely linear increase. This is really interesting because it suggests a negative feedback on UBE3A expression when genomic overdosage starts to get really, really high, as you would see, especially in this UBE3A+4 case. So in other words, there might be a limit to how much UBE3A you could possibly overexpress in the brain. So this might have implications for interpretation of further studies. So our model clearly overexpresses UBE3A, but is it expressed in the right place? That's what we wanted to test next. And to figure this out, we stain for UBE3A protein in the brain, which you can see here in the top panels of this slide here, clearly it has a green fluorescent signal. And if you focus on the top panels here and over here, you can see that UBE3A protein is virtually ubiquitous in neurons throughout the wild type brain. So if you look front to back, top to bottom, you see UBE3A in neurons anywhere you look. Contrast that with what you see in the Angelman syndrome model mice, which carry a maternal UBE3A deletion and thus express no UBE3A in their mature neurons. It's virtually a blank slate. And we took advantage of this by crossing them to our UBE3A+2 overexpressors, thereby allowing us to evaluate the overexpressed protein alone in isolation to see if it in and of itself was trafficked and expressed according to endogenous patterns. And so what we saw is that although there was certainly a lot more of it, the transgenic protein distribution does look just like wild type. And this is true whether you're looking broadly across the brain in sort of a zoomed out fashion, or if you zoom into specific regions or even specific neurons and neuron types, it looks very similar in terms of biodistribution to the wild type, the native state. So that leads us to, collectively these data support that our model is suitable for addressing a very important question, which is, what is the phenotypic impact of UBE3A overexpression? And where does one start when you have a model like this in hand? Well, considering our expertise and how severely seizure conditions can impact the quality of life, we chose to focus first on seizures. So seizures in Dup15q syndrome can be severe and debilitating and affect up to 63% of individuals. And they do appear to be worse for IDIC-15 individuals than for individuals with interstitial Dup15q. Moreover, SUDEP, or sudden unexpected death in epilepsy, is also a serious concern. And again, it's more so for the IDIC-15 individuals. So basically, we're hoping that we can leverage our UBE3A+1 and +2 models to help us determine if there's a similar dose-dependent effect of UBE3A overexpression in mice that kind of mirrors what we're seeing clinically with respect to seizure phenotypes. So to explore this, we subjected both UBE3A+1 and +2 mice to the Flurothyl kindling model of epileptogenesis, which is schematized here. So this may look complicated, but it's a rather simple assay in which mice are exposed to a vaporized proconvulsive drug called Flurothyl until they have a seizure. And we primarily just score the time it takes for this seizure to manifest. So on day 1 in our schematic, the mice are naive. And so any information we get on that day kind of speaks to their baseline seizure susceptibility. But then through repeated once daily exposures to Flurothyl over the next 7 days, or so-called kindling, we're gradually lowering the seizure threshold in these mice. So in other words, the mice are having seizures faster and faster each day, and this suggests that their brain circuits are becoming increasingly hyper-excitable. So similar to what happens in epilepsy and the brains of people with epilepsy. So the permanence of this change really speaks to a mouse's susceptibility to epilepsy. And we test this by exposing the mice to Flurothyl once more after a full month off without any Flurothyl exposure. We give them one more challenge and see if any changes induced by the kindling are permanent or not. So to give you an idea of what a pro-epileptogenic phenotype looks like, I'll quickly walk you through some data from one of our previous studies involving Angelman syndrome model mice. So plotted on the x-axis here is the Flurothyl trial day, which I just explained to you in the schematic. And plotted on the y-axis is the seizure latency. So specifically just the time it takes for a mouse to exhibit a seizure once Flurothyl is delivered. And as you can see, while Angelman syndrome mice, which are represented by the plotted green line here, they show a modest reduction in seizure threshold on day 1. There's really no difference in their kindling slope. But nevertheless, upon re-challenge, so after that month off that I explained, they are exhibiting a dramatically heightened sensitivity to Flurothyl, which is consistent with their having an enhanced propensity to develop epilepsy. So this is an assay that we use to probe our UBE3A overexpressor model. And when we did so, we looked at the UBE3A+1 and +2 mice in the exact same experimental paradigm, but somewhat surprisingly, saw no evidence of enhanced seizure susceptibility. So it didn't matter what specific manifestation of the seizure we looked at, whether it was myoclonic twitching or jerking, or a full-out generalized seizure in which they completely lost posture, and in some cases exhibited tonic-clonic manifestations, or on the day we looked. So it didn't matter if we looked on day 1, throughout kindling, or during the re-challenge. We saw no difference between our UBE3A overexpressors or the wild-type control mice. So these data suggest that UBE3A overexpression alone is insufficient to increase seizure risk or to exacerbate epileptogenesis. And moreover, possibly that other overexpressed genes in the 15q11, q13 region may co-contribute to seizure phenotypes in Dup15q syndrome. So this is an intriguing hypothesis that we're eager to follow up on. That said, what did become obvious during the course of these experiments was that both UBE3A+1 and UBE3A+2 mice were far more likely than control mice to die from Flurothyl-induced seizures when they did happen. So we found this to be actually very surprising. So the death in the control group was pretty rare. So fewer than, roughly 5 to 10% at most were succumbing to seizures during 8-day kindling. And compare that with at least 35% of UBE3A+1 mice dying, or greater than 50% of UBE3A+2 mice dying during this 8-day course of kindling. So this phenotype, we think, it's very striking, and we think it might have implications for SUDEP in Dup15q syndrome. Further studies will, of course, be required to see if there's really something to this, and we're currently pursuing those in earnest. But of course, seizures account for just one of the many phenotypic manifestations that are relevant to Dup15q syndrome. And with the remainder of our time, my colleague Matthias is going to communicate to you a summary of his beautiful work detailing the performance of the UBE3A overexpression model that we created in various other molecular and behavioral assays. So with that, I'll turn it over to Matthias.
All right. Do you see the proper screen here, or do we need to swap displays? Can anyone confirm?
I see it. Okay.
Okay, great. Okay, well, hello everyone. My name is Matthijs, and I'm a PhD student in the lab of Overexpression at the Erasmus MC in Rotterdam. And I'll continue the talk and show you more of the behavioral analysis and characterization that we did of this overexpression mouse model that Matt just described. And I'll end off with some even more extensive molecular analysis that we did. Well, just very extensively described this Ube3A overexpressing mouse model that was generated in the Philpot lab now harbors 2 extra genetic copies of the UBE3A gene. And we obtained that mouse also at Erasmus MC and used it as a start point for our experiments, wherein we generated 3 different types of offspring of this mice. So we have a wild type mouse, which is mostly the control, a Ube3a+2 mice that has 2 extra genetic copies of the UBE3A gene, and then the Ube3a+4 mice that have 4 extra genetic copies of the gene. And this is a nice way to kind of titrate out the dose of Ube3a and see whether there are dose-dependent effects or results of Ube3a overexpression on behavior. And firstly, before we commence with any experiments, any behavioral experiments, we tried to validate that indeed on the protein level, also Ube3a was nicely overexpressed. So we looked into several regions of the brain. So we looked at hippocampus, cortex, striatum, and cerebellum. And what you can nicely see is that when you look at here in the y-axis, the Ube3a abundance, that with increasing genetic copy load, also the protein amount really goes up. So that was a very nice starting point for our behavioral experiments, where our main aim is to behaviorally characterize these mice to study the effect of Ube3a overexpression. Well, before I want to go into the specifics of the behavioral test that we did, I first want to take a small step back and saying that we at the Overexpression lab are very much interested and have a lot of expertise on Angelman syndrome, which actually occurs when, as Matt said, you have a deletion of the Ube3a gene. And over the years, we have published several studies where we describe the establishment of a behavioral test battery, where we test or where we were able to distinguish wild-type mice from AS mice, so mice that are deficient in Ube3a. And how does that look? Well, this behavioral test battery actually comprises of 5 different tests, wherein we see differences when we compare wild types with Angelman mice. So if we look at their capabilities to walk on a rotating rod, or the amount or the distance that they walk in an open field, the amount of marbles that they bury, the amount of nest material that they use, and their willingness to swim in a forced swim test actually is constantly impaired or decreased in Angelman mice. So we figured that these tests show some sensitivity to the Ube3a level, because if Ube3a is not there, the performance in these specific tests is impaired. And with this reasoning, we thought, well, if Ube3a is maybe overexpressed, we might find similar phenotypes in these tests. But for the results of that, I can actually be very short or brief, because in neither of the genotypes we found any significant impairments in performance in these tests. So all the mice were able to perform equally well as the wild types, showing that Ube3a overexpression has no general effect on the performance in these different tests. Now, of course, the question is, how relevant are these specific behaviors to something like Dup15q syndrome? And that's also, of course, the question that we had. So we wanted to dive a bit more into the specifics of Dup15q syndrome. Therefore, we took a look at what is the symptomatology that goes associated with Dup15q syndrome, which is intellectual disability, hypotonia, epilepsy, dysmorphic features, and autism spectrum disorder. And we tried to translate that into something that we could measure in our mice. So we looked at learning and memory. Matt just showed you his data on epilepsy experiments, and we also looked into social behavior. Well, first we'll show you the experiments that we did on learning and memory. And in this first experiment, we exposed our mice to a fear conditioning test or paradigm. In this test, we place a mouse into a novel context, which is a box that has a metal grid on the floor. During the first 150 seconds of the trial, we just measured the activity of the mice. And mostly they explore the box a lot, so they hardly freeze, and they're mostly moving. And then after 150 seconds and also 30 seconds later, they are administered a light foot shock through the metal wires on the floor, which leads them to associate this context, this box that they're in, with the fear memory. So the moment they're put back 24 hours later, or even 28 days later for long-term memory, mice that have remembered this well will show a lot of freezing behavior because they will anticipate this food shock. And that is what we quantified here in the graphs. So here you see that on baseline, in the 3 different genotypes that we compared, so the wild type, the +2 and the +4 mice, there is actually hardly any freezing. However, after the conditioning, after day 1, but also after day 28, there is a lot of freezing happening. But when we compare the 3 different genotypes with each other, we actually do not find any significant differences. Meaning that in the sphere conditioning paradigm, UBE3A overexpression does not seem to have so big of an influence. Well, as an extension to the learning and memory paradigms that we looked at, we also performed a Morse water maze test. And in this test, we place our mice into a big circular water maze where a hidden platform is located here in the TQ, which stands for target quadrant. And the rest is divided in Q1, 2, 3, which is just 3 other quadrants. And during a trial, a mouse is placed in this bath, and this platform they cannot see whilst they're swimming, but they only notice when they swim over it that they can stand on it, and it prevents them from having to swim. But they have to learn to locate this platform based on external cues outside of the maze. And they learn this through 7 days of consecutive training. And then on the 8th day, we actually measured the locomotion behavior to look at where their preference is, where they're mostly trying to swim to find this platform. And you can see already from the heat maps that most of the mice swim around the target quadrant trying to find back this platform, meaning that they do have learned where and how to locate the platform. But then again, when we compare the 3 different genotypes with each other, it doesn't seem that UBE3A overexpression has any effects on the spatial memory acquisition that these might obtain. Well, that was for the learning and memory. We also performed, as I said, experiments to assess social behavior, and we did this by means of a 3-chamber test. And in this test, we place our mice into a big box that is divided into 3 subcompartments. So one containing a cup that has a novel mouse under it. We have a center compartment, and we have a novel object containing compartment, which basically is just an empty cup. And we measure the time the mice spent in either of the 3 compartments that is expressed as here in the opaque bar of the wild type around the novel mouse, in the center, or around the novel object in the stripe bar. And what you would like to see is that if a mouse displays social behavior, it would spend most of its time around the novel mouse, which is also what you see here for the wild types. But when we compare it to the UBE3A+4 mice, we actually see really the same pattern, where there's also an increased time spent around the novel mouse. And when we compare the sociability preference, there actually seems to not be a difference. And as an extension to the sociability test, we also tested social novelty, where now under the formerly empty cup, we place a novel mouse. And we'd like to see a shift of attention towards this now newly introduced mouse. And that's also what you see here in D, where now the attention is shifted towards this novel mouse. But the same thing is observed in the UBE3A+4 mice, meaning that again, it doesn't seem that UBE3A overexpression in these mice, in this specific test, has any effect on the sociability of this mice. Well, that's mostly it for the behavior. Then lastly, we also had a look at molecular effects, and specifically at the transcriptome. Since we know that UBE3A might have some effects on specific transcripts, and actually it might function as a transcriptional co-regulator. We isolated the brains of 5 wild type and 5 UBE3A+2 mice and looked specifically into the cortex in the hippocampus at an age of 7 days after birth. So really during the process of neurodevelopment still. And we performed an RNA sequencing on the tissue. And firstly, we analyzed the data by means of a principal component analysis. And what you do with this analysis, you try to see whether there is clustering of the different groups that you kind of send out to see if there's a difference between the groups. And what you can see in this graph is actually that there is clustering of the hippocampus and the cortex. So the origin of the tissue we can find back in this analysis. However, we fail to find any separation when we look at the wild types or the cortex, meaning that they're actually very much alike. And this is also even more clear in this following plot here in B, where we find that UBE3A gene is really the only transcript that is significantly altered in its expression, whereas the other genes that we identify here do not pass the thresholds of significance. So that brings me to the conclusion in saying that, well, in these mice, UBE3A overexpression is not sufficient to induce the behavioral or transcriptomical phenotypes. As Matt showed, elevated UBE3A levels seem to increase the likelihood of seizure-associated deaths. But the remaining question, and actually also something Matt already kind of addressed in one of his last slides, is probably UBE3A might be involved in any of these phenotypes, but might not be a singular. There might not be a singular role for UBE3A, but other genes and proteins that co-contribute with UBE3A, eventually culminating in this Dup15q symptomatology. Well, remains for me only to thank Matt and Ben and all the other colleagues at the University of North Carolina for a very fruitful collaboration. And at Erasmus MC, my students and the technicians who helped out a lot, Ube3a+ for my supervision, the committee of the Moving Mountains for having me and organizing this event. And I want to thank you all for listening and your attention. I'd be very happy to take any questions.
Any questions?
Thank you for opening to questions. As a mom, I'm looking at the learning in the non-wild type mice and how visually they look like they have significant differences and differences over time, where initially, I don't know how to describe it, but I know it. I know studies. I know you're supposed to look at significant difference, but I'm looking on the water maze test, the difference, and I'm not sure, I guess, not being familiar with the test or the study, what the Q3, Q1, and Q2 refer to, but it looks like over time, it becomes easier for them to remember, but initially it's harder to remember. It reminds me of the way my daughter learns that sometimes things hold, or sometimes they don't hold and they come back later all of a sudden. And I've heard this from other parents, that things look like they're not there and all of a sudden they're there. And if you could, is there a way you could increase the stress in the test to see? I almost wonder if the stress of learning it, you know, if the death comes from this connection of UBE3A to the phenomenon of cell apoptosis, if there's something about UBE3A that's triggering death of cells in a seizure event, and a seizure event is similar to a learning event because it's this excitatory event, is there a possibility that something about the excitatory nature of learning would cause it to be more difficult for the mice to remember in that Q2, or what was before that? Before the water maze, you had another test of learning and memory. It just, yeah, see, the chamber test, I'm looking at that, you know, it's just interesting to see how much lower the +2 mice are than the wild type mice, and to see that there's a substantial, a visually more apparent increase in freezing behavior in UBE3A+4 mice. I'm thinking that increased fear conditioning, if it's attracted to that increased fear conditioning, that fear conditioning is a very high energy emotion. And so you've got a lot of stimulation going on there in the neurons, and whether that something about that excitatory nature in the similar way to the excitatory nature of a seizure. Sorry, that's an overly complicated question.
Yeah. I was like, so would you like them to respond, or do you want to answer your own question?
No, I'm not answering my own question. I'm wondering, I want to know if they think that's connected to an excitatory nature of learning.
Okay.
That's what I want to know.
Okay.
Do you want to take this, Matthias?
Yeah, for the specifics, I wouldn't have a very good answer. with regards to the point that you make about the difference that you might see here between the wild type and the UBE3A+2 mice, I guess you do have a point in saying that it might appear a bit higher, and of course we base all our conclusions mostly on statistical tests, and now we see that this is not significant, but also there's a lot of spread within the data. So I guess that also mostly explains why these differences are actually not picked up with any significant tests.
I think one kind of broader theme that I think was touched on in the question was, whether, there might be ways you could change the assays, maybe to challenge the mice more, make the task more difficult in different ways, whether that's, putting a fear component into it, or maybe even something with the Morris water maze. So Matthias mentioned that there are spatial cues around the arena, right, that they have to use to locate the escape platform. And so maybe if those are a little bit less salient, you might tap into something. So I don't think we could I would just say, I don't think we could, another way to put it is, I don't think we could conclude definitively with any of these assays that there isn't, an impairment to the neurodevelopment of the circuits that's imposed by there being more UB3A there, but we just may not have teased it out all the way, and based on the design of the assay, and that if you did, overexpress other genes in concert, other 15q, 11q, 13 genes in concert with UB3A, maybe that would be another way to sort of eclipse the threshold for, a phenotype. So, there could be a lot going on there. And maybe another way to think of it too is, UB3A may need help. So I think Matthias put it very nicely with his last slide. It may need help to get you over the threshold for seeing some of these phenotypes, but that being, it may also be, the overexpression may be absolutely necessary for any of them to occur as well. So it's very complicated. I think we're just on the cusp of starting to maybe use these models to tease these things apart. And so I think it's a very complex biology, and I think your question reflects that. I think it's appropriately complex. It's just the state of things now.
We're going to do one more question, and then we're going to go to Stormy. So
just wondering if you could talk a little bit about the negative feedback that's silencing UB3A overdosage and causing that sublinear response. I don't know if you can speculate as to some of those mechanisms.
I have only just the most nebulous ideas about it myself. I frankly was not expecting to see anything like that at the RNA level necessarily. You could imagine a number of transcriptional feedback mechanisms, either directly or indirectly, that might kind of attenuate accumulating overexpression as you increase gene dosage. I think a lot of people, based on the auto ubiquitination and capacity for auto degradation at the protein level, I think we would have expected to see it at the protein level, and I think we are seeing that. But to think that there's an RNA level of feedback regulation perhaps as well is also fascinating. And I don't know, maybe Matthias has more concrete ideas about a mechanism, but I think it's definitely something we want to follow up on, but I don't know if I have anything specific.
No,
oh, sorry. Go ahead, please. No, you're the one who has the answer.
I don't really have anything more to add to this. I would say indeed now it seems that, aside from the fact that I always had the impression that UB3A only would auto-regulate itself on the protein level, now we have this additional auto-regulating role on transcriptional level, where potentially even transcription factors that are necessary for the transcription of UB3A itself might be in some way targeted by UB3A, which is all pure hypothetical, of course. But I guess
just real quick, is the naive assumption that you would now just start stacking other critical genes on top of this mouse, since you seem to have put them through their paces through all these tests and don't really see any difference? Would you now just sort of go candidate gene by candidate gene stacking on top, like overexpressing HER2 now, or is that kind of the naive progression?
I don't think it's naive. I think it's labor intensive. I think you would want to be really carefully consider the cost benefit analysis of any of those endeavors, because they won't be trivial to pursue. But no, I think that's a very interesting direction to take this research. And there might be other ways to sort of get at that as well, whether it's in induced iPSC-derived neuronal models. Maybe there's some phenotypic traction there where you can do similar things genetically with a little easier capability maybe than mouse modeling. But yeah, the general thread of thrust of your question is well taken. I think that there's definitely a lot of intrigue there.
Thank you.
So I know that there's more questions, but if you can put them in the app, they will get them and then they can respond to them too, because we're already a little bit over. But a big thank you to Matt and Matthias, and thank you for staying up for us. Great.